Welcome back to the deep dive. Today we are tackling a really massive shift in how we think about infectious diseases.
It really is. We're sort of moving away from the old world and into something completely new.
We're moving away from the petri dish and really into the genetic code itself.
You know, it's a fundamental change in mindset. There's a specific smell that goes with traditional microbiology. I mean, if you've ever walked past a hospital lab or even just a college bio lab, you know what I'm talking about.
Oh, yeah.
It's that earthy, yeasty, sometimes slightly sweet, sometimes absolutely, revolting smell of things growing.
The smell of the agar plates, the sheep blood agar, the chocolate agar, all of it.
Exactly. It's the smell of life. And for over centuries, since the days of Pasteur and Koch, that smell was the foundation of diagnosis. It was what I like to call microbiological farming.
Microbiological farming, I like that.
Yeah, you take a clinical sample sputum, blood, urine, and you sow the seeds onto an agar plate. And then you wait. You wait for the harvest. You wait for that colony to grow big enough to be visible to the naked eye.
And today, what's the new model?
Today, we are moving full speed toward microbiological forensics.
Forensics.
Yep. We aren't waiting for the bug to grow anymore. We are hunting for the fingerprints it left behind at the scene of the crime. We are looking for its DNA, its RNA, or its very specific protein signatures.
And that is the absolute core premise of today's deep dive. We're going to break down Chapter 5 of Murray's Medical Microbiology, which is all about molecular diagnosis.
And we're going to layer in some of the clinical nuance from Chapter 47 of Jollitt's.
Right. Our mission here, for you, is to build a solid mental model for how we identify these pathogens without ever seeing a single colony.
And for the learner, I mean, especially if you are prepping for exams, this is incredibly high-yield territory. The examiners absolutely love this stuff.
Why is that?
Because it sits right at that perfect intersection of basic science mechanism. You know, how does the test actually work? And direct clinical application. When do I order it? And more importantly, what do I do with the result?
Right. It's not just about knowing what PCR stands for. It's about knowing why you'd use it for a viral load, but might, hesitate to use it for a test of cure.
Yeah.
There's a big difference there.
Precisely. And we need to get very clear on the difference between detecting the organism itself, which is what we're talking about today with molecular methods,
versus detecting the host response.
Right, which is serology, a whole different can of worms. And we also need to understand the evolution, the shift from these slow, old-school biochemical identification methods to the lightning speed of things like MALDI-TOF.
Okay, so let's get the lay of the land for this discussion. I think we have 4 main stops on our roadmap today.
Sounds good.
First, we'll start with what I'm calling the detective's magnifying glass, which is all about probes.
Okay.
Second, the Xerox machines. This is where we get into the big amplification techniques like PCR.
The heavy hitters.
Yep. Third, we'll cover reading the blueprint sequencing and figuring out exactly who the culprit is. And finally, the protein revolution, MALDI-TOF.
It's a very logical progression. We start simple, just looking, and then we get progressively more high-tech as we go.
Let's do it. Section 1, the detective's magnifying glass. We're talking about non-amplified nucleic acid probes. Now, probe is kind of a vague word. What exactly is a probe in this context?
A probe is it's essentially a tool for finding a very specific sequence of DNA or RNA. So imagine you have a single strand of DNA from a pathogen. That strand has a unique sequence of letters A, C, T, G.
The genetic code.
The genetic code. A probe is just a short synthetic strand of DNA or RNA, we call it an oligonucleotide, that is designed to be the exact complement to that pathogen's unique sequence.
Okay, so if the pathogen has a sequence of, say, AATTGC.
Then the probe is designed with the sequence TTACG.
And when they meet, they bind.
They bind. They hybridize. I like to think of it as molecular Velcro. The probe is the hook side, and the pathogen's DNA is the loop side. If the pathogen is present in the sample, the Velcro sticks. It's that simple.
But just sticking isn't enough, right? I mean, you need to know that it's stuck. You can't see a single DNA strand with your eyes.
Correct. And that's why the probe always carries a tag. It's like a fishing hook with a little glow-in-the-dark bobber attached to it.
So you can see it.
So you can see it. The tag might be a radioactive isotope, though we use those a lot less now for obvious safety reasons. More commonly, it's an enzyme that can trigger a color change or a chemiluminescent or fluorescent molecule.
So the hook sticks, you wash away all the unattached probes, and if there's any signal left, you see the light.
That's it. You found your target.
This sounds great. I mean, it's specific, it's direct. But Murray throws in a pretty big caveat right at the start of the chapter. He says these probes, on their own, usually lack sensitivity for direct clinical specimens. Why is that? What's the catch?
The catch comes down to numbers. It's a signal-to-noise problem. Let's say you take a sample of sputum from a patient with possible pneumonia. In that sample, there might only be a few hundred, maybe a few thousand bacteria. So you throw in your probe. You might get a few hundred or a few thousand of those little hooks to stick.
And a few thousand microscopic glowing dots.
It's completely invisible. It's just not enough signal to be detected by our standard instruments against all the background noise of the gunk in the sputum sample.
So the signal is just too weak.
It's too weak because we haven't amplified anything yet. We're just finding, and there's not enough to find.
So if you can't really use it directly on the patient's sample, where does it fit in? What's the clinical use case?
And this is a really crucial exam anchor. These probes are absolutely ideal for identifying organisms after they have already grown in culture.
Ah, okay. So we're back to farming for a second. We're not escaping the agar plate entirely.
Briefly, yes. You need it to bulk up the numbers. Let's say you've got a slow-growing colony on a plate. It looks like some kind of mold, maybe a fungus.
Right.
Now, it could be a harmless environmental mold, or it could be a dangerous dimorphic fungus like histoplasma or coccidioids. Traditionally, identifying that could take days, even weeks, of looking at structures under a microscope and doing biochemical tests.
A long, anxious wait for the patient and the clinician.
A very long wait. But because you have a physical colony, you don't have a few hundred bugs. You have millions of bugs. You have a massive amount of target DNA.
So you can just take a little scoop of that colony, put it in a tube, add the probe.
And because there are millions of targets, you get a massive, bright, unmissable signal.
So it's not for primary diagnosis, but for rapid confirmation.
Exactly. It confirms the identity almost instantly. It's very, very common for identifying things like Mycobacterium tuberculosis or those tricky fungi. It replaces days of tedious work with a simple, definitive yes or no genetic test.
Now, Jawetz highlights a specific type of probe technology that feels like the pro version of this. It's called PNA-FSH.
Right.
Peptide nucleic acid fluorescence in situ hybridization. That is a mouthful. Let's break down the chemistry here, because Jawetz says it's the chemistry that explains the clinical value.
It's a really fascinating bit of bioengineering. So standard DNA has a sugar phosphate backbone. And if you remember your basic chemistry, phosphates are negatively charged.
Right. Anions.
Right. So since both the probe DNA and the target DNA have these negative charges all along their backbones, they naturally repel each other slightly.
Yeah, trying to push the north poles of two magnets together.
That's the perfect analogy. They resist binding. It takes energy and time to overcome that repulsion and get them to stick together.
And PNA, how does that change things?
PNA probes are synthetic. They're man-made. Scientists went in and completely replaced that charged sugar phosphate backbone with a neutral peptide backbone, which is basically a protein backbone.
So no negative charge.
No charge, no repulsion. Which means the PNA probe binds much, much faster and much tighter to the target DNA than a natural probe ever could. It's like super Velcro.
And why does this matter clinically? I mean, why do we need super Velcro? What's the rush?
The rush is because it allows the test to work in much messier, more complex environments with fewer cleanup steps. And that enables a very specific, very high-yield workflow, particularly with positive blood culture bottles.
Okay, set the scene for us. This sounds important.
It is. Imagine a patient is septic. They're in the ICU, high fever, hypotensive, really sick. You draw blood cultures. 12, maybe 14 hours later, the machine in the lab beeps. The bottle is positive. Something is growing.
The clock is ticking.
The clock is absolutely ticking. You pull some fluid out of that bottle, you do a quick gram stain, and under the microscope you see gram-positive cocci in clusters.
So it's staph. But is it Staphylococcus aureus, which is a potential killer? Or is it a coagulus-negative staph, a con-NS, which might just be a skin contaminant from the blood draw?
That is the million-dollar question. And usually you'd have to take that fluid, subculture it onto an agar plate, and wait another 24 hours to see if the colony turns gold or to do a coagulus test. That's 24 hours of clinical uncertainty.
And 24 hours where the patient is probably on broad-spectrum, heavy-hitting antibiotics like vancomycin, just in case.
Exactly. PNA-FSH lets you skip that wait. It's sensitive enough that you can take a drop of fluid directly from that soupy, dense blood culture bottle, apply the PNA probe, and in a couple hours, you know for sure if it's a psoriasis or not.
That is huge for antibiotic stewardship. You can de-escalate or stop the vancomycin if it's just a contaminant.
Huge. And Jowetts also mentions its use for Candida, differentiating the common Candida albicans from some of the more drug-resistant non-albican species like Candida auris. It's all about getting actionable information with speed and precision right when it matters most in critical care.
Okay, so to sum up this first part, probes are great for finding a target if you have a lot of it, like in a colony or a positive blood culture. But what if you don't? What if you are looking for that classic needle in the haystack? A tiny amount of virus in the blood, or just a few bacteria in a urine sample.
Then you need a bigger machine. You need Section 2, the Xerox machines. We are moving from simply finding the target to nucleic acid amplification, or NAA.
The goal here shifts completely. We aren't just trying to find the needle in the haystack anymore.
Yep.
We're trying to duplicate the needle over and over until the entire haystack is made of needles.
That is a great analogy. It's exactly what we're doing. And the undisputed king of this technology, the one everyone has heard of, is, of course, polymerase chain reaction, PCR.
The thermal dancer, as you called it.
Yes, because PCR relies on cycling temperatures to drive a chemical reaction. It's a three-step dance that happens over and over again. And to really get this for an exam, you have to be able to visualize what is physically happening to the DNA at each of those temperatures.
Okay, let's walk through the choreography. Step 1 of the dance.
Step 1 is denaturation. You take your sample, which has the DNA in it, and you heat it up to about 95 degrees Celsius.
Nearly boiling.
Pretty much. This intense heat is enough to break the weak hydrogen bonds that hold the DNA double helix together. The two strands of the latter just unzip. They separate, creating two single strands.
Step 2.
Step 2 is annealing. For this, you cool the mixture down pretty significantly, usually to somewhere between 50 and 60 degrees Celsius. This cooler temperature allows your primers to find their target and attach.
And primers are absolutely key here. What exactly are they?
Primers are short, specific, single-stranded DNA sequences that you, the scientist, have designed. They act like bookends. They are engineered to match and bind to a very specific sequence on the pathogen's DNA that you're looking for, and they mark the starting point for copying. They essentially tell the enzyme, copy the bit between here and here.
And then step 3.
Step 3 is extension. You warm it up again slightly, this time to about 72 degrees Celsius. This happens to be the optimal temperature for the workhorse enzyme, DNA polymerase specifically, a heat-stable one called Taq polymerase.
Right.
The polymerase latches onto the primer that's already bound to the DNA, and it just starts adding the building blocks, the A's, C's, T's, and G's, filling in the rest of the strand and creating a new complementary strand.
So you started with one double-stranded copy, and after one cycle of heat, cool, warm, you now have 2.
Exactly, and then you just do it again. Denature at 95, anneal at 55, extend at 72. 2 copies become 4, 4 become 8, 16, 32. It's exponential amplification.
2 to the power of n, where n is the number of cycles.
Precisely. If you run 30 cycles, which takes maybe an hour or two in a modern machine, you go from a single starting copy to over 1 billion copies.
That's the power of PCR. That's how you can detect an incredibly small amount of a pathogen. You don't find the signal, you manufacture the signal yourself.
That's a perfect way to put it.
Now, for the exams, it's not enough to just know standard PCR. That's table stakes. We need to talk about the different variations, the different flavors of PCR.
Right. These are the high-yield differentiators. First up, and maybe the most important variation, is RT-PCR.
Which stands for reverse transcriptase PCR.
This is critical to understand. Standard PCR, as we just described it, works on a DNA template. But many of the viruses we care about most, HIV, influenza, hepatitis C, and of course COVID-19, are RNA viruses. Their genetic material is RNA, not DNA.
And DNA polymerase can't read RNA.
It can't. It's the wrong language, so you need a translator. In the first step of the reaction, you use an enzyme called reverse transcriptase. This enzyme reads the RNA template and synthesizes a complementary DNA copy, which we call cDNA.
Okay, so you're turning the RNA into DNA first.
You are, and once you have that stable cDNA, the rest of the process is just a standard PCR amplification. So the RT part is that initial conversion step.
So mental check for the listener. Reverse means going backwards from the central dogma from RNA to DNA before you start the main amplification.
Exactly.
Okay, next variation. Nested PCR.
Nested PCR is all about achieving ultimate precision and sensitivity. It uses 2 separate rounds of amplification, back to back.
How does that work?
You run a normal PCR with 1 set of primers. Then you take the product of that reaction, all the little DNA copies you just made, and you use it as the template for a second PCR. But this time you use a new, second set of primers that are designed to target a sequence inside the piece of DNA you just amplified.
So it's like putting a target inside a target. You have to hit the first one and then the second one.
Yes. And that dramatically increases both sensitivity and specificity. Because think about it. If the first set of primers accidentally bound to some random non-target DNA and amplified it, the chance that the second, internal set of primers will also find match on that random piece is astronomically small. It weeds out the false positives.
Very clever. Then we have the one I see in the hospital all the time on order forms, multiplex PCR.
Yeah.
The respiratory panel or the meningitis panel.
Multiplex is all about efficiency. Instead of running one test for one bug, you put multiple different primer pairs into the very same reaction tube.
So you're hunting for multiple suspects at once.
At the same time, in the same sample. For a meningitis panel, in one tube of cerebrospinal spinal fluid, you might have primers for Streptococcus pneumonia, primers for Neisseria meningitidis, primers for Haemophilus influenza, and primers for enterovirus.
And if any of those are present in the sample?
Their specific primers will find their target and trigger amplification. You get a result for everything at once. It's a huge time saver.
And finally, the one that has really become the modern standard in most labs, real-time PCR, which is also known as qPCR, for quantitative PCR.
Right. So in the, let's call them the old days of the 90s, after you finished your 30 cycles of PCR, you had to do a whole other step. You had to take the tube, open it up, pipette out the liquid, and load it onto an agarose gel.
And then run electricity through the gel to see if you got a band of the right size.
Exactly. It was slow, it was messy, and it was a huge source of problems. Real-time PCR gets rid of all that. It measures the amplification as it happens in real time.
Wow.
You add fluorescent dyes or probes into the reaction tube from the very beginning. Every time a new copy of DNA is made, it generates a little burst of fluorescence. The machine has a detector that watches the amount of fluorescence increase with every single cycle.
So you can literally watch the curve of the signal go up on a screen.
You can. And this brings us to the big exam point that both Murray and Joetz emphasize. Why is real-time PCR so much better and safer for the laboratory environment?
It has to be because you don't open the tube.
It's a closed system. You never, ever open the tube after you start the reaction. In the old gel-based days, when you opened those tubes, you were releasing a cloud, an aerosol, of billions of microscopic DNA fragments, we call them amplicons, into the air.
Which would float around the lab.
And land on your workbench, on your pipettes, on the next patient sample you're setting up, causing massive contamination and false positive results. Real-time PCR almost completely eliminates that risk of amplicon carryover. It's cleaner, safer, and faster.
Okay, that covers the thermal cycling methods, the dancers. But not all amplification needs a heater and a cooler. Let's talk about the isothermal engines, the ones that work at a single constant temperature.
These are very clever. They use enzymatic tricks to amplify nucleic acids without the need for a thermal cycler machine. And the most important one clinically, the one that Joetz really highlights, is TMA transcription-mediated amplification.
Joetz calls this the gold standard for, and I'm quoting here, the clap and the chlamydia.
He's not wrong. It's the technology behind the most common tests for Neisseria gonorrhea and C. trachomatis. TMA is different because it primarily amplifies RNA, not DNA. It uses a cocktail of enzymes, including RNA polymerase and reverse transcriptase, to simulate the natural biology of transcription. It's a self-sustaining cycle that produces billions of RNA copies very, very quickly, all while sitting at one constant temperature.
And because the target is RNA, which is less stable than DNA, it's incredibly sensitive for detecting active living organisms.
That's a key advantage, yes. Then we have SDA strand displacement amplification. Another isothermal method.
This one has a cool mechanism.
It does. SDA uses a special kind of trick. It uses a restriction enzyme to nick the DNA. That means it cuts just one of the two strands.
Okay, so it doesn't cut all the way through.
No. Then a specific type of DNA polymerase comes in. It sees the nick and starts to synthesize a new strand using the intact one as a template. But as it builds the new strand, it acts like a snowplow. It physically pushes the old nick strand out of the way.
It displaces it. Hence the name.
It displaces it.
Yeah.
And that displaced strand is now a new single-stranded target for more primers to bind to and the whole process to repeat. It's a continuous chain reaction at one temperature.
I love that snowplow metaphor. And finally, there's one more isothermal method that has a very, very cool visual trick. LMP,
Ah, yes, loop-mediated isothermal amplification. LAMP is fascinating, if a bit complicated. It uses a set of 4 to 6 different primers that are designed to recognize multiple regions on the target DNA.
And they do something strange to the DNA structure, right?
They do. They cause the amplifying DNA to loop back on itself, creating these complex cauliflower-like DNA structures. It's an incredibly rapid and efficient amplification process.
But the visual part, the trick.
So as this massive amount of DNA is being synthesized, the reaction produces a huge amount of a chemical byproduct.
Yeah.
Magnesium pyrophosphate.
Now, what is that?
It's an insoluble salt. It's a white precipitate. So as the reaction proceeds, the liquid in the test tube literally gets cloudy. It goes from clear to turbid.
So you don't even need a fancy fluorescence detector. You can just look at it.
In some settings, yes. You can just hold the tube up to the light. If it's cloudy, it's positive. This makes it an amazing technology for low-resource settings, for point-of-care testing. It's used for rapid TB testing in areas without big, expensive labs. We also use it in hospital labs for things like C. difficile testing.
Okay, so we've covered magnifying the signal with probes, and we've covered Xeroxing the signal with amplification. Now we need to move to Section 3, reading the blueprint, nucleic acid analysis.
This is where we stop just asking the question, is it there? And we start asking exactly, who are you?
And the absolute star of this show, the workhorse for bacterial identification, is the 16S rRNA gene. It's often called the barcode of bacteria.
It's a perfect name for it. Every single bacterium has ribosomes to make proteins, so every bacterium has the gene for the 16S component of the ribosome.
But what makes this one gene so special for identification?
It's the structure of the gene itself. It has some regions that are conserved, meaning the DNA sequence is identical or nearly identical in almost every bacteria on the planet.
That's handy. Gives you a universal landing pad for your primers, right?
Exactly. You can design a set of universal primers that will bind to and amplify the 16S gene from any bacteria you happen to find. But, and this is the key, in between those conserved regions, there are 9 hypervariable regions.
These are unique.
These are unique to the species, or at least the genus. The sequence of A's, T's, C's, and G's in those variable regions is like a fingerprint.
So let's use a clinical scenario. You have a patient with a brain abscess. The neurosurgeon drains the pus. You send it to the lab, but nothing grows in culture. It's a classic case of mystery meat.
That's the perfect scenario for 16S sequencing. You take that pus, you extract all the DNA from it, you use your universal primers to copy whatever 16S gene is in there, and then you send that product for sequencing.
You read the letters.
You read the letters of the variable regions, you get a sequence back, you plug that sequence into a massive online database like GenBank, you hit search, and a second later it tells you, this sequence is a 99.9% match for Fusobacterium nucleatum.
And now you've solved the unsolvable case. You know what you're treating.
It has revolutionized how we handle culture-negative infections.
And for fungi, is there an equivalent?
There is. The concept is identical. We just use a different target gene. For fungi, we typically use the 28S rRNA gene or regions called the internal transcribed spacers or ITS regions. Same idea, different barcode.
Now, 16S identifies the species. But sometimes, in an outbreak, we need to go deeper. We need to know if the salmonella making people sick from lettuce in California is the exact same strain as the one popping up in New York. For that, we need whole genome sequencing, WGS.
WGS is the heavy artillery of public health and epidemiology. Instead of looking at just one gene, which is about 1,500 letters long, we look at the entire genetic code of the bacterium, all 4 or 5 million letters of it.
And you're comparing the genomes from different patients, letter by letter.
Exactly. We're looking for tiny differences called SNPS single nucleotide polymorphisms. So if you have 2 bacterial isolates, one from California and one from New York, and their entire 4 million letter genomes differ by only 2 or 3 SNPs, they are virtually identical. They came from the same source. That tainted lettuce was shipped nationwide.
But if they differ by hundreds of SNPs.
They are completely unrelated. It's 2 separate outbreaks. WGS gives you an incredible level of resolution for tracking infections.
This high-resolution approach has largely replaced the older method, right? I'm thinking of RFLP restriction, fragment length polymorphism.
Yes. RFLP is the old guard. It's worth knowing for historical context and because it still pops up on older exam questions. RFLP was a much cruder way of comparing genomes. It involved using molecular scissors.
Restriction enzymes.
Right. These are enzymes that recognize and cut DNA at very specific sequences. So the idea was, if two bacteria had slightly different DNA sequences, the scissors would cut in different places.
And you'd get DNA fragments at different lengths?
Exactly. You'd then separate those fragments by size on a gel using electrophoresis. The smaller fragments move faster, the larger ones move slower. You'd end up with a unique pattern of bands for each strain. It looked a bit like a supermarket barcode.
And there's a special version of this for really big pairs of DNA, right?
Yes. For trying to compare entire bacterial chromosomes, you needed a technique called pulsed field gel electrophoresis, PFGE. A huge DNA molecule is like a long, tangled piece of spaghetti. It just gets stuck in the gel matrix and can't move.
So what did pulse field do?
PFGE cleverly changed the direction of the electrical field, back and forth, side to side. This forced the big DNA snakes to constantly reorient and wiggle their way through the gel pores. It was the gold standard for outbreak tracing for decades, but WGS just gives us so much more detailed, unambiguous data now.
Okay, let's shift gears completely. We've been talking nonstop about DNA and RNA, the blueprints, but there's been a revolution in the lab that ignores nucleic acids entirely. It looks at the final product, the proteins.
Yes,
Yeah.
the Melditov manuscript.
That's quite a name. Matrix-assisted laser desorption ionization time-of-flight mass spectrometry. That sounds like something straight out of science fiction.
It really does. But it has completely, and I mean completely, transformed the day-to-day workflow of the clinical microbiology lab. It is a true game changer.
So let's visualize this for the listener. Walk us through it. You have a bacterial colony growing on an agar plate. What do you do?
You take a sterile toothpick, you touch the colony, and you take a tiny, almost invisible smear of the bug, and you spot it onto a metal target plate. Then you pipette a tiny drop of a chemical called a matrix on top of it.
What's a matrix?
It's usually a simple organic acid. It mixes with the bacteria, and as it dries, it co-crystallizes with all the bacterial proteins trapping them.
Okay, bug on a plate with some crystal matrix.
Yeah.
Then comes the laser.
Then comes the laser. You slide the plate into the machine, it pulls a vacuum, and it zaps the spot with a laser beam. The laser energy is absorbed by the matrix, and it transfers that energy to the bacterial proteins. This does two critical things at once: desorption and ionization.
Desorption means it blasts the proteins off the plate and into the air, into the vacuum.
It vaporizes them, and ionization means it gives them a positive electrical charge. So now you have a cloud of charged protein molecules floating at the starting line of a race.
And then they fly.
They fly. There's an electrical field that accelerates them, and they shoot down a long vacuum tube toward a detector at the other end. This is the time of flight part of the name.
And the physics here is actually incredibly simple, isn't it?
It's high school physics. Force equals mass times acceleration. If you apply the same accelerating force to all the charged particles, the small, lightweight proteins will accelerate very quickly. They'll fly fast. The heavy, bulky proteins will accelerate more slowly. They'll fly slow.
So the detector at the end of the tube just records the time it takes for each protein to hit it.
Exactly. It creates a spectrum, which is a graph with a series of peaks based on the mass-to-charge ratio, millimeters d, of each protein. It's a unique spectral fingerprint for that organism.
What proteins are we actually looking at here? What makes up this fingerprint?
It's primarily ribosomal proteins, and that's for a very good reason. They are incredibly abundant in the cell, and they are evolutionarily stable and conserved. They make for a very reliable, reproducible signal.
So the machine generates this spectral fingerprint, and then it compares it to a massive library of known fingerprints.
And in literally seconds, it spits out an answer. This spectrum is a 99.9% match for Pseudomonas aeruginosa, identification complete.
So why do students, and more importantly hospital administrators, care so much about this technology?
Two words: speed and cost. A traditional biochemical identification where you put the bug in 20 different little tubes of sugar and wait 24 to 48 hours to see which ones turn yellow is slow and requires a lot of expensive reagents.
Right.
Melditov gives you an answer in minutes, and the reagents for one spot cost, literal pennies. The machine itself is expensive, but the per-test running cost is negligible. It has saved labs and hospitals a fortune.
But there's a catch, isn't there? There's always a catch.
There's always a catch. The main limitation is that you usually need a pure colony to start with. It doesn't typically work well if you just smear raw sputum on the plate, because you get a messy mix of proteins from many different bacteria plus human cells.
So you still need the farming step first.
For most applications, yes. though there are now FDA-approved protocols for using it directly on positive blood culture fluid, which, again, speeds things up tremendously.
This brings us to our final and maybe most important section, Section 5. We have all of these incredible, powerful tools, PCR, L&D, WGS. But, powerful tools can be dangerous if you don't know how to interpret the results correctly. We have to talk about the so what.
This is the most critical section for any future clinician. You absolutely cannot simply read a lab report as positive or negative without putting it into the full clinical context of your patient.
And the first big trap here is what Murray and Jatchowitz both refer to. It's that confusion around the dead bug effect.
Right. Let's be very clear. PCR detects nucleic acid. It does not and cannot distinguish between nucleic acid from a live replicating organism and nucleic acid from a dead fragmented one.
DNA is just incredibly stable.
Yeah.
It can stick around in the body for a long time after the organism that made it is long gone.
For weeks, sometimes. So if I treat a patient for chlamydia with a course of antibiotics and they come back to the clinic 2 days later for a retest,
the PCR will almost certainly still be positive.
it will. Not because the treatment failed, but because the PCR is just amplifying the ghost DNA, the remnants of the bacteria that were successfully killed by the antibiotics.
So high-yield exam tip. You do not order a molecular test of cure immediately after treatment for things like C. difficile or chlamydia. You have to wait for the body to clear out all that dead material.
You have to. Otherwise, you'll misinterpret the result and overtreat the patient. This leads directly to the second major concept of interpretation: colonization versus infection.
This is a huge one. Just because the DNA is there, it doesn't automatically mean it's the cause of the disease.
The absolute classic example of this is Clostridioides difficile. A PCR test for C. diff looks for the gene that codes for the toxin, like PCDB. It detects the potential for the bug to cause disease.
It does not prove the bug is actually producing the toxin.
Correct. So if you have a patient in the hospital who is perfectly healthy, no diarrhea, but you do a screening PCR and it's positive, that patient is simply colonized. The bug is there, but it's not doing anything. The positive result is, in that context, clinically irrelevant.
Another example they mentioned.
Aspergillus. If you do a bronchoalveolar lavage on an ICU patient and the PCR comes back positive for Aspergillus DNA, what does that mean?
It's ambiguous.
It's very ambiguous. Is it a true life-threatening invasive aspergillosis, or did the patient just happen to breathe in a common harmless Aspergillus spore from the air and you're just detecting that one little piece of DNA? You can't tell from the PCR alone.
You have to correlate it with the patient's immune status, their CT scan, other biomarkers. The test is just one piece of the puzzle.
It's one piece of the puzzle. Finally, let's touch on resistance markers. This is a growing application. We can use PCR to detect the gene for resistance, for example, the MEK gene for MRSA, or the VNA gene for VRE.
And the benefit there is speed. You can put a patient into isolation precautions immediately without waiting 2 days for culture-based susceptibility results.
It's great for infection control, but you always have to remember genotype does not always equal phenotype. Just because the gene is there doesn't mean it's being expressed. And more importantly, bacteria can develop resistance through new mutations that your targeted PCR test won't even be looking for.
So it's a good screening tool, but the gold standard is still growing the bug and actually testing it against the antibiotic.
The phenotype is still king.
All right, we have covered a massive amount of ground. From simple probes to sequencing to flying proteins, I think it is time to consolidate all of this.
Let's do it. Time for some rapid recall.
Okay, so I'm going to ask a question based on everything we've just discussed. I'll pause for a moment, and you, the listeners, should try to answer it in your head, and then our expert will give the correct answer and a brief explanation.
Ready when you are.
Question 1. A lab technician is performing an amplification assay. They notice that the reaction tube, which started out clear, has become visibly cloudy due to the precipitation of magnesium pyrophosphate. Which amplification method is being used?
That would be LMP, or loop-mediated isothermal amplification. That turbidity is the classic visual hallmark of a positive LMP reaction.
Perfect. Question 2. You need to rapidly identify a slow-growing gram-positive organism that is just flagged positive in a blood culture bottle. To do this, you use a special synthetic probe with a neutral peptide backbone that allows for faster, tighter binding. What is this technique called?
That is PNA-FEAISH, peptide nucleic acid fluorescence in situ hybridization. The neutral backbone is the key to its speed and utility in that critical setting.
Excellent. Question 3. In a Maldi-Toff mass spectrometry analysis, What specific cellular component is primarily being ionized and measured to generate the organism's unique spectral fingerprint?
The primary targets are the ribosomal proteins. They're used because they are so highly abundant, stable, and conserved, which gives a very strong and reproducible signal.
Great. Question 4. A patient is treated for a chlamydia infection with appropriate antibiotics. 2 days after finishing the treatment, a follow-up PCR test is still positive. Does this result definitively indicate treatment failure?
No, it does not. PCR is detecting residual nucleic acid from dead bacteria, which can persist for days or weeks after successful treatment. It is not a good test of cure in the short term.
Very important point. Question 5. A physician suspects a patient has an acute infection with an RNA virus, such as influenza or HIV. Which specific PCR variation must be used to detect this type of pathogen?
That requires RT-PCR, or reverse transcriptase PCR. You must first use the reverse transcriptase enzyme to convert the viral RNA into cDNA before amplification can begin.
And for our last question, question 6. From a laboratory workflow and safety perspective, what is the single greatest advantage of using real-time PCR, or qPCR, over the older traditional gel-based PCR methods?
The primary advantage is that it is a closed system. Because the tube is never opened after the reaction starts, it drastically reduces the risk of amplicon carryover contamination, which can lead to false positive results.
Fantastic. To really solidify all of this, we strongly encourage you to review Murray Chapter 5, especially the diagrams on PCR cycles and the MALDI-TOF workflow, and to check out Jowett's Chapter 47 for those great clinical examples.
And before we sign off, maybe one final thought to leave you with.
Please.
We're moving towards a future of metagenomics where we don't even target one bug. We just sequence everything in a patient sample directly. The DNA of the patient, the bacteria, the viruses, the fungi, all of it.
So a lab report of the future might list 50 different organisms found in a single sputum sample.
Exactly. And the future physician's challenge won't be finding the bug. It will be wading through that sea of data and deciding which of the 50 bugs we found actually matters, which one is the pathogen, and which are just innocent bystanders.
So as the technology gets better and more sensitive,
the clinical art of interpretation, of knowing the patient, becomes more important, not less.
a perfect place to end. We'll see you in the next deep dive.